Hello everyone. We are students of class ninth performing an activity on preparing a stained temporary mount of onion peel. Material required for this activity are an onion, a cover slip, a glass slide, a needle, forceps, glycerine, a compound microscope, blotting paper. Now the procedure to do the activity. Cut the onion first in half and then in quarter pieces lengthwise. Take one of the quarters and separate the fleshy leaves from the stem. Pull out the thin transparent layer from the concave or inner part of the fleshy leaves with the help of forceps. Put it on the watch glass containing water. Cut the peel into small square pieces. Add a drop of iodine to the watch glass containing onion peel. Select one piece and transfer it to the slide. Add a drop of glycerine to the peel and cover it with a cover slip. Observe the slide first under a low power and then high power. Now the observation for the activity is the cell of onion peel are rectangular shape. Each cell is bounded by distinct cell wall. Rounded, deeply stained body is observed in every cell. This is called nucleus. There may be one or two large central vacuoles. The lightly stained substance toward periphery is called cutoplasm. Now conclusion for the whole activity. As the cell walls and large vacuoles are clearly observed in all the cells, the cell placed for observation are plant cell. Onion epidermal peel is made up of rectangular shaped cells. A nucleus, a central vacuole, a thin layer of cytoplasm, and a cell wall make up each cell. Since each cell has cell walls and large prominent vacuole, the cell under observation are plant cell.